the quantity of cancer biomarkers present within the human body. So that's, again, a very elegant example of utilizing the concept of magnetic relaxation switches using magnetic nanoparticles. Moving from very innovative ideas to solve biological problems, very interesting concept is the tissue engineering. This is magnetic force-based tissue engineering. Obviously, again, magnetic nanoparticles are involved. Here you have liposomes with magnetic nanoparticle core. And these liposomes are connected to, say, for example, stem cells. So when such stem cells bound to liposomes are utilized for agglomeration on a surface with a magnetic field underneath, what happens is there is a layer-by-layer assembly, a very simple and elegant layer-by-layer assembly of different layers of these stem cells. And when you remove the magnet, you can clearly isolate such sheets of stem cells. And apparently, such sheets of stem cells have been extremely effective in bone tissue engineering. The results have been compared with the bone tissue engineering made with such tissues in comparison with the free cells. And bone tissue engineering made with such tissues have been extremely good. The last example I would like to share with you is again a very elegant design. And this design consisting of a 3-in-1 system for magnetic resonance imaging, optical imaging, and photodynamic therapy. The nanoparticle design here is a magnetic core silica shell nanoparticle. And the silica shell consists of an iridium complex. And that complex is responsible for singlet oxygen generation, which is necessary for the photodynamic therapy. And this is also responsible for optical imaging. So such systems have been found to be very useful in cancer treatment. So having given you few examples as to how one can manipulate the size, the shape, and the composition, I would like to move on to the next stage of opportunity that we have to manipulate the magnetic nanomaterials, that is magnetic modulation using oscillating magnetic field. So this brings us to a next level of opportunity that we have for utilizing the magnetic nanomaterial. I would like to share with you 2 examples here. I think by now we are already familiar about thermotherapy, hypothermia using magnetic nanoparticles. And in fact, there is a company, MagForce Nanotechnologies. It's a German company which is in the final stages of clinical trials for treating glioma. So this is a very good example of using hypothermia to treat cancer. The next example I would like to share with you is remotely triggered release from magnetic nanoparticles. This is, again, a very interesting concept. The concept is based on generation of heat on application of electromagnetic force. So in this case, you have a DNA molecule bound to a magnetic nanoparticle, and a complementary DNA molecule is attached to complementary molecule, which is either a drug or a targeting agent or a diagnostic element, is attached to this particular construct. And when you apply an oscillating magnetic field, say about 4 kilohertz, this bond gets destroyed and releases this particular moiety. And this has been shown to be very successful even in vivo studies. I would strongly recommend you to go through this paper published in Advanced Materials last year. And this is a very elegant design. I mean, on one side, we keep hearing about magnetically modulated approaches, but this particular approach is really elegant. So now I move on to share the design that we have been working on in the past 4 or 5 years in my laboratory. And this particular design is a very ambitious design, and I would like to really call it as more a magic than a reality at this point of time. So what is this design that we are trying to develop? The questions that we asked were, can we protect the drug till it reaches the target? So the previous design that we have seen here, even though it's elegant, you would see that this drug moiety is exposed to the external environment. So that's not really desirable in biological milieu. So we were looking at trying to develop a system, design a system where the drug is protected till it reaches the target site. The next question we asked ourselves is, can we have multiple guidance and therapy? Many times what happens is you have a design element where you have a single guidance and a single therapy. And many times such single guidance is not really successful. So having an opportunity to have a multiple guidance and multiple therapeutic elements present in your design offers you a foolproof treatment options. The third question that we asked ourselves was, can we have remote control release? Remote is very important, and control is very important. The control release is important because we are not only trying to release the drug in a controlled manner, but you want to release it at the target site. So it has a combination of guidance and control release. And we also would like to release the drug however much we want at whatever time that we would like to release. So that's the key to answering this question. And of course, finally, can we have simultaneous diagnosis and therapy? I have no doubt in my mind that we are moving towards personalized medicine. The concept that most of the pharmaceutical companies follow, which is bulk drug concept. That is, you are trying to have a drug that is applicable for 7 billion people on this planet. And that's not sustainable at all. So we are going to move, if not today, in the next decade or so, into the personalized medicine. And this is key to the success in personalized medicine, having an opportunity to simultaneously diagnose and treat. So keeping in view that all these elements, having all these elements present in your design is key to successful magnetic modulated integrated diagnosis and therapy, we went on to design our nanoparticles. And here, what we looked at was LHRH as a targeting agent. LHRH is luteinizing hormone and releasing hormone. And this particular hormone has receptors at least in 15 different types of cancers. And they are present in, these receptors are present in very large numbers on these cancers. The most prominent among the cancers in which the LHRH receptors present are breast, ovarian, and so on. So we decided to utilize the LHRH as a targeting agent. And then we need a magnetic element, and we need a drug that is protected. Therefore, you have a drug that is encapsulated within a polymeric material, and then you have magnetic nanoparticles, again, within a polymeric nanoparticles. So when you have such a design, one could, in principle, have a multiple targeting. The multiple targeting comes from LHRH, number one, and number two is through magnetic targeting. And you can have a multiple therapy. So you can release the drug using oscillating magnetic fields. You can also use hyperthermia. So it's a two-pronged approach for treatment. So having designed such a multifunctional nanoparticle, we went on to demonstrate various elements present in this design. So what are the various elements present in this design? First is, of course, to demonstrate that it's possible to have multi-targeting and therapy. Next is to design magnetic nanomaterials. One is to design and optimize the magnetic properties in order to ensure that there is an optimum release of drugs within this polymeric nanostructure. The next element, very important element, is to demonstrate remote control release. And finally, it is important to demonstrate that such a construct we can also utilize for diagnosis. So let me give you some background on what's the drug that we have chosen. We have chosen lytic peptides as the anti-cancer drugs here. Lytic peptides are actually the next generation anti-cancer drugs. These are membrane disrupting peptides, and they have been shown to be extremely potent against breast cancer cells. As I've already mentioned, breast cancer cells express receptors for LHRH. So it is also known through our collaborators' research work at Pennington Biomedical Research Center that LHRH, when you combine it with the lytic peptide, this particular conjugate has a site specificity thanks to the LHRH ligand, and it has a therapeutic lytic peptide. So the only problem with a simple LHRH lytic peptide conjugate is that it also goes to gonads. The gonads have obviously the receptors for LHRH. Therefore, this particular treatment is, while it's effective, but it has its own side effects. So this is where our design is very effective. Now, just to say a little bit more about the LH ligands, they're really unique because not only LHRH ligands have receptors on breast cancer cells, the ligands themselves prevent reticuloendothelial uptake. And why is it so important? If you look at various nanoparticle designs, all those successful designs have PEG linkers. The reason why you have the PEG linkers is just to prevent RES uptake. And in our case, we have the LHRH not only acting as a targeting agent, targeting moiety, but also it's preventing RES uptake. Such a design, we have demonstrated, I'm not sharing the results with you, is that you can counter the diffusional instability as well. So having this basic information, our design is supposed to work this way. You have the functionalized nanoparticles with a magnetic core, and you have the polymeric shell containing the lytic peptide conjugates, so for example, Hecate, beta-CG, or you can even take just the simple Hecate, which is a lytic peptide. And you have the LHRH ligands on the surface of these polymeric nanoparticles. And such nanoparticles are anticipated to dock onto the breast cancer cells. And when you apply an oscillating magnetic field to such construct on a breast cancer cell, there is a release of drug. And this is the concept that we started working on. So the first thing that we did was to design appropriate magnetic nanomaterial. And herein we utilized what is called as a core shell structure. So the magnetic properties can be varied very elegantly by using this particular approach. So when you vary the size of the core and the size of the shell, you pretty much vary the magnetic properties. And you, in fact, get magnetic properties which are better than when you vary the composition of a single metallic nanomaterial. And that's what we have seen by having cobalt core copper shell and cobalt core gold shell nanoparticles. And then we went on to see whether really applying an oscillating magnetic field would help us to release these drugs in a controlled fashion. So to demonstrate this particular concept, we designed nanoshells based on manganese carbonate core and what is called as a layer-by-layer self-assembly approach. And when you design these micron core nanoshell particles with dispersed cobalt core and gold nanoparticles, and such design, when such nanoparticles have been taken into FITC solution, a fluorescent solution, we see that when you apply an oscillating magnetic field, the fluorescent dye permeates through these empty core nanoshell materials. Whereas when there is no magnetic field, there is no permeability of fluorescent dye within the empty core shell. So this clearly demonstrates that one can modulate the release of drug or any material by manipulating utilizing the oscillating magnetic fields. And in this particular case, we have utilized the cobalt core and gold shell nanoparticles. And we also would like to rule out that such a release is not based on the heat that's generated, rather than it is truly based on the oscillating magnetic field. So when we took these particular constructs in an FITC solution and heated the whole solution to 50 degrees centigrade, and we did not see any permeability at all of these nanoshells. So that clearly rules out the possibility that permeability is happening because of the heating. We have also looked at the influence of the size of the bilayers or the size of the shell. And we clearly see that there is a limitation on the size of the cell that is really important in order to optimize the design for a controlled release. We have also looked at the magnetic resonance imaging of LHRH spions. And we are really happy to see that when you have LHRH spions in comparison with the pure spions without any LHRH agent, there is an increasing amount of iron oxide nanoparticles within the cancer cells. And we could also see, this work is done in collaboration with Warren's group in Duke University. I'm not an expert in MRI, but what I hear is that you can clearly distinguish several features when you have LHRH spions in the magnetic resonance imaging of not only the breast tumor, but also the metastatic cells arising out of the primary breast tumor. I understand that the hyperpolarized helium MRI, in fact, helps in increasing the sensitivity of these lung metastatic cells, and one could clearly distinguish up to the micron size various metastatic cells that are present in the tissue. So what I'm going to do is I'm going to stop here. I know we are all waiting to finish this conference, and my time is also up. But as I mentioned, I think I just left this as a part of my presentation because if someone is really interested, they can go through it, and I'll be happy to discuss this. The whole concept is about developing the nanotechnology tools for pharmaceutical industry, and I'm going to leave it here. So the take-home message is that magnetic nanomaterials offer unique opportunities in medical diagnosis and therapy. And there are really broad range of nanotechnology tools that are available. The range is inexpensive to expensive, most elegant to simple nanotechnology tools. And I truly believe that the personalized medicine is the way to go. This is the strategic tool that we are trying to develop, which is the IBIN, Innovation Box for Implementation of Nanotechnology Tools. Is a useful guide for strategic implementation of nanotechnology tools. And MagNano can provide assistance in implementing such tools in pharmaceutical industry. So I would like to thank you for your attention, and also, of course, collaborations are key to be successful in nanotechnology area. And I've been very fortunate to have wonderful collaborations from Duke University, Bonn University, and of course, people from LSU and CAMD itself. Thank you.